So guys, today the first student is going to do a presentation, which is going to be Paolo, which is my classmate. We are the 6th year of MD program, and Paolo hopefully will be a future neurosurgeon. And today he's going to talk about something really complicated to me. So, okay. Single-cell RNA sequencing to study T-cell gene expression for Neo-Antigen Vaccine in Glioblastoma. So difficult to read. Okay. So guys, I think you will start and enjoy. Okay, we can start. Be late, but again, enjoy.
Thank you. Good evening, guys. We made it. All right. So today's presentation is about a work that I did this summer when I was in Boston. I'm interested in neurosurgery and especially brain tumors. And this summer I did some research on a new immunotherapy method to try to solve the problem that is still glioblastoma multiforme. Okay, so let's get started. So, okay. Not working, of course. Okay. So single-cell RNA-seq is a very complex, very expensive technique, which is getting very trendy in molecular biology these days. What it basically does is tag, sequence, and analyze RNA transcript from single-cell suspensions. And in this way, you can trace back which set of RNA molecules comes from every single specific cell that you have analyzed. So this technique is already really powerful for normal cells, but it reaches its pinnacle of power when you analyze cancer cells. And the reason for that is that it combines mutation profiling of cancer cells. So it tells you which cancer cell has which specific set of mutations. And at the same time, it analyzes the transcriptional profile. So which set of genes are upregulated and downregulated in every single specific cancer cell. You combine these two information together, and what you get is no longer just the definition of cancer cell identity, so which tumor cells we're dealing with, but it's a much more hidden, more fluid, very active and dynamic definition of cells. So cancer cell states. So this already tells you the hidden activity of cells as they do their job, as they proliferate, which pathways are activated, the mutations that they have, the potential therapeutic resistance profile, and we can know all of this for every single cell that we analyze in a tumor. Now, back to GBM. So I may be a nerd for what concerns this field, but when we speak about glioblastoma, we're not just dealing with maybe the most untreatable and lethal tumor in the body. We're dealing with something that is so much more. You know, we've been looking at this thing for 60 years now. We tried every possible therapy. None of it is working. Patients keep dying within a 1-year lifespan. So we have no idea of what's going on until single-cell analysis and multi-omics techniques came around. So now we actually know something about this tumor. We know why this is so biologically complex, and we know why it is so difficult to treat. And we know that there are 3 rules, 3 basic layers of complexity that explain the therapeutic hardships and the biological profile. So rule number 1 is GBM tumors are so complex because of the cells themselves. So there is no single cell that has the same mutation profile than the other, and the transcriptional activity is even more fluid. The second point of complexity is my favorite, is glioma stem cells. So for now, the spoiler is every single tumor cell can put, no matter the differentiation background, can potentially be a stem cell. So that's all that I'm going to say for now. And then finally, the kaleidoscope of normal cells that are exploited, hijacked to do the tumor cell's bidding, the tumor microenvironment. So let's first start with tumor cells themselves. So what do I mean when I say that there is overlap between tumor cells and stem cells? Okay, so if you take a sample of GBM tissue and you run single-cell RNA-seq on this, the final result that you see is this compass-like kind of cardinal orientation in the gene expression map. So there are 4 tips, 4 cellular orientations that identify cellular states. These are combination of genetic signatures. So which genes are upregulated and downregulated. The set of combination of upregulation and downregulation is a signature. The combination or group of all the signatures of a cell is its cell state. And we now know that in GBM, cells can be of 4 different cell states. 3 of them recapitulated the neurodevelopmental lineage. So for instance, neural progenitor cells, oligodendrocytes, and astrocytes. So these tumor cells kind of look like the healthy counterpart of the brain tissue. On the other hand, mesenchymal-like cells are not similar to the normal brain tissue. They're more similar to stromal cells. The cool point about all of this is that cells do not just rigidly fall into one of these 4 categories. Here comes the concept of glioblastoma as the shapeshifter, mask changer. Every single cell, no matter the original state, can freely shift between all the other 3. And if you implant a single cell of a given cell state in a culture, it can not only change its cellular state into any of the 4 options, it can generate cells of every other cellular state, no matter the initial one. So, and then we got stem cells. Okay, so the breaking news here is that glioma stem cells have been puzzling the field for around 10 years. And the reason for that is in a Science 2017 paper, it has been shown that differentiated tumor cells at some point under some specific genetic clues or some microenvironmental cues, at some point they just overexpress neurodevelopmental transition factor and they de-differentiate. They turn into stem cells, even though they were fully differentiated. So this is the game changer. This is not the Yamanaka experiment. There are no researchers throwing transcription factors at cells in culture. This happens lively in nature and in truly pathological tissue. So the corollary idea is, what is a glioma stem cell? Well, the shortcut answer is everything and nothing, because in this way, every single tumor cell can potentially be a stem one and generate the whole tumor entirely. That's why, even though you do a gross total resection, it doesn't really matter. You leave one cell, the whole tumor can be generated. No matter if it is still differentiated, these cells differentiated, they turn into stem cells, and they start it all over again. But we do have markers that define glioma stem cells. And it is actually been shown that there are different markers according to the original stem cell state of the cell, as you can see here. The whole point is it doesn't really matter because we don't have a transcription outlook for glioma stem cells, and the tumor markers change when the cell state changes. So again, we don't have a clear answer for what glioma stem cells are, what's their contribution, how we can stop them. And if this wasn't crazy already enough for you, there is the tumor microenvironment. 10 different cell types, normal cell types that are exploited by the tumor in order to boost its own proliferation. So I divided these cells into 2 groups. The old one, the one that are more described in the literature and the one we know more about, are called the old guards and are here represented in red. On the other hand, those cells that are more experimental in the sense that we are working with them towards achieving new therapeutic perspectives, but those that we don't really know much about, these are called the new entries, and here are represented in red. So the first old guards that we're going to describe today are tumor-associated macrophages. So the shortcut explanation is that macrophages are recruited by the tumor in order to do tumor immune suppression. So any kind of therapeutic that is immunotherapy based fails because of tumor-associated macrophages. There are 2 cell populations, the bone marrow derived macrophages that penetrate into the into the brain as a result of the inflammatory reaction that the tumor generates, and microglia. Microglia are present in the brain tissue from the fetal development, but nonetheless, they can contribute. And although they are supposed to be very different in theory, in practice, this is not true. As you can see here in this graph, so again, the red in these graphs is upregulated genes, the blue is downregulated genes. And you can see that the gene expression profile for microglia kind of like fades where the macrophage expression profile begins. So again, this tells you about the concept of fluidity of active cells between one state and the other, both in the normal cells, but especially in pathological cells, or normal cells like tumor-associated macrophages that are under the influence of the tumor. You all thought that neurons were just the offended part? Wrong, they are not. They are the traitors. They are the mutineers on board. They help tumor cells proliferate. How? By hooking them up into neural circuits and exploiting the synaptic transmission to stimulate cell proliferation pathways within glioma cells. This is another game changer. Have a look here. It doesn't really matter whether the synapse is chemical or electrical. Both are able to boost and upregulate the proliferation pathways within glioblastoma cells. Glioblastoma cells may choose to make use of the neuron either directly or through the help of stromal cells, thereby boosting and expanding their echo of influence in the brain tissue. And this is at the whole system level. So at some point, the tumor may even be able to induce enough brain dysfunction to alter the immune stress response. Why is that? So in the hematopoietic stem cell niche in the bone marrow, some key regulators are also beta-adrenergic nerves that are in place. And when there is a strong stress from the body, they release synaptics openly into the bone marrow niche in order to start mobilizing cells and produce new white blood cells. So this is also the reason why if you are very stressed, for instance, you may have transient period of immune suppression. This is part of the explanation. So imagine what would happen if a tumor, consciously or not consciously, would cause such a strong brain dysfunction that it would alter also this pathway of immune response regulation. We would lose the battle from the very beginning, no matter which immune therapy, chemotherapy we can give. There is no prepared immune response to deal with the tumor in the first place. Of course, we got the poor good guys, the T cells. They are very strong, very powerful. Their anti-tumor immune response is very specific, but unfortunately, they have to deal with, their angry brothers, the T regulator cells, microglia, macrophages, and also have a look at this. These are the poor T cells, and the blue ocean is all the tumor cells. They're like the 300 Spartans in front of the Persian army. They're like, ok, raga, facciamo quello che possiamo, non garantiamo niente. So, but nonetheless, they are very powerful. Their specificity and specific anti-tumor immune response is very promising, and we hope to make use of that to kind of tackle the tumor in its status right now. And this is where part of my work comes in. So the basic concept behind a T-cell vaccine is you take some tumor tissue, you dissociate it, you take some tumor proteins, and then you inject them into the patient along with some myeloid cell booster. So the basic idea is that antigen-presenting cells can uptake those proteins, present them to the tumor cells become activated against them, they travel all the way to the circulation, they cross the BBB, and finally they reach the tumor and target them very specifically. That's the theory, at least. And the T-cell vaccine that I worked with this summer kind of follows this idea. So when the tumor first starts, so the primary tumor is first taken out, dissociated, you analyze tumor cells, and then what do? You basically look at neoantigens. So neoantigens are mutant proteins expressed only by the tumor. For this reason, the body is more likely to recognize them as non-self. So they are more immunogenic. So this is good. So you further increase the specificity and the immunogenicity by selecting into these neoantigens some 50 to 30 amino acidic peptides that are predicted with some prediction algorithms to bind even more specifically MHC class 1 molecules of the patient. So after you have isolated this very important super immunogenic, super personalized set of peptides, you select 15 or 20 of them, and you make 4 different pools for possible cocktails, and you inject them into the patient. And this is for the primary tumor right after the surgery. So at the recurrence, when the tumor has recurred, because it always does, what you do is you take it out again, you do the surgery, but you discard the tumor cells and you focus on the T cells. What we are interested in is understanding whether those T cells that were educated for these epitopes actually are specific in their TCR expression profile for those antigens that we injected. So that's what I did. Basically, this is my small contribution there. I used the SMART-Seq2 protocol for single-cell RNA-seq on T cells that were extracted from the tumor at recurrence. This protocol starts with what we call a whole transcriptome amplification. So you take the RNAs from single-cell suspensions, so they are isolated into single wells, so they know that all the RNAs from that well are from one single cell. And what you do, you basically convert them into DNAs and you do their sequencing with PCR. You need to make them very specific. You need to add a tag that tells you, okay, all these transcripts comes from these specific cells. So what do you do? First, to these DNA molecules, you add adapter frames, nucleotide sequences, and then you had first a forward primer of 5 nucleotides and then a reverse primer of 7 nucleotides. Now you can take and merge all the RNA fragments together because you know that they are tagged. They are very specific and you know where they come from. So this is what I did. You make some final pooling, and then you do a quality control. And after that, after 6 weeks, analyzing one single patient, 10 96-well plates, using 180 boxes of tips, not just single tips boxes, you merge everything into a single Eppendorf, not the whole rack, just the little guy, 20 microliters for 6 weeks. Exciting. So after that, you see. So this is the sequencer, you put it in and you kind of. So I didn't get to see the final result because I'm not so. This part I have 11 minutes. It's gonna be okay. There are 7 patients and it's gonna be vaccine last issue. But it tells you that there is something. We have much more knowledge. We know the best immunotherapeutic strategies for this tumor, and we're just trying because there's no other way around. I'm going to leave you with something interesting, a new perspective on this tumor is spatial transcriptomics. So if you look at this sample, what if you were able to do single-cell RNA-seq, but not in cell suspensions, retaining information of the position of cells in the tissue? So here you got clusters of RNA-seq, but also you cannot see where its position is in the original slide. This is really important because it can tell you very specific information from the local microenvironment. It can tell you what was the original cell state in a portion of the tumor. Was there any specific type of microenvironment there? Can we get any more cues, anything that can help us for the treatment? And yeah, that was my last slide. I thank you for your attention and I look forward to your questions.
Thank you, Paolo. Thank you, Paolo. So guys, if you have any question, Paolo will be glad to answer, I guess. Also online, if someone has a question.
I like it's a really correct way it's formulated. Well, there are cases in which either glioblastoma itself or its metastasis may the MHC1 may downregulate MHC1 molecules and end up being MHC1 negative. In these cases, does this vaccine provide any kind of secondary function of some other cell line, for example, NK, immune function that can provide an effect also in these kind of cell populations.
Right, so that's the limitation of this vaccine, because you have trained the T cells for a specific set of peptides that you knew were holding on for the tumor, the primary one. You don't really know whether they're still the same, they're still there at recurrence. That's why we're interested in doing this analysis. We want to know whether these T cells are still proliferating when they infiltrate the tumor. This would tell us, okay, those antigens are still there and the vaccine is still effective. But of course, being so fluid and so plastic, GBM cells can just change them in a second. So yeah, the efficacy is still limited as the protocol is today.
Anyone else, guys?
During the personalized choice of what's the risk of autoreactivity?
So basically, the risk of autoreactivity is very low because we are specifically selecting neo-antigens. So these are proteins that are mutated only in the tumor. So you have the risk of cross-match is actually very low.
Maybe more related to therapy in general rather than your specific project. But do you think that coupling this vaccine together with the current chemotherapy could help further the activity of immune cells against the tumor, maybe with the release of
Well, yes, potentially, because chemotherapy is intended for direct tumor cell killing. So the more antigens and proteins the tumor cells release, the more likely it is that some immune cells may pick up just by chance, because of course the immune cell penetration in the brain is very low. But when you have something as inflammatory as a tumor, nonetheless, some cells keep crossing the blood-brain barrier. The blood-brain barrier itself is dysregulated, it is somehow broken. So of course, yeah, chemotherapy can help in this. Sorry, say it again. Okay, this is nice. Well, I think that for now, this has been attempted only for GBM. And the reason for that is that other lower grade tumors are not really immunogenic. The T-cell profile in oligodendroglioma and IDH mutant glioma has been analyzed, but unfortunately, especially IDH mutant expresses a molecule that is called R2HG. This is actually a catabolite from an alteration in the Krebs cycle. This molecule, nonetheless, has been associated with a further immune deficiency, immune suppression. So T cells do not really pick up on lower grade gliomas per se. If you add this metabolite, the response is even poorer. So for now, this will only be applied to GBM. Also for lower grade gliomas, it's easier not to get to immune therapy because you know that surgery and some chemo would work better.
So my it was kind of similar, but I was also wondering whether about the other types of vaccines that are being developed in a similar way, but maybe targeting I don't know, some more shared interpersonally antigen. So objectively, this is a very, very personalized way to make a vaccine. And when brought to the clinical practice, it means a very high cost of production for the vaccine. And of course, a limited use in the sense that, of course, we're not speaking about the uber common thing, but still, maybe feasible probably in very, very high specialized centers, but not available to a global scale. So maybe, do you know about some research that tries to find some molecules that are more, let's say, share with your person and can you more formulation compatible way?
Yeah, that's a good question. So I know about a study in 2019 that did this for IDH mutant glioma. The results, as I said, were not enthusiastic because, of course, the immune response is even poorer than GBM, as we said. And getting to glioblastoma itself, it wouldn't really work because, as we've seen, the fluidity of cells between one state and another is so intrinsic that they could potentially express thousands of proteins, but at different time, at different moments, the proportion in the whole tumor would be different from person to person, and it would be different in the same tumor over time. So it's not really standardizable at the moment, but it would be nice to do, for instance, serial vaccinations. Some serial treatments are actually attempted at this moment, and this could, help reassess the genetic profile, the neoantigen profile of the tumor. But again, you're working against time because we know that even though you have chemotherapy, surgery, immune therapy, these patients still die within a year and a half at best. So it is still work in progress.
Any other question?
I think I have it here. Let's see. Yeah, right here. You administer the vaccine before, after radiotherapy, because of course radiotherapy increases cell damage and potentially new antigens that you could tag and target, and right after surgical resection. So I guess chemo works the same. It has not been reported in the original paper, but again, you can do this in combination.
Okay, any other question? This is the last time I'm asking. Okay. Okay, thank you for coming today. Thank you very much, Paolo, for being our first student to present. We hope to see you all in the second semester. We still do not have dates. No. We still don't have dates. Abbiamo un altro incontro? Purtroppo no.
No.
Niente spritz. Dobbiamo fare. So we hope to see you in the second semester. We all wish you good luck for the exams and also Merry Christmas.